2.A.2. You need to stop talking now, guys. I don't understand why. I think you messed with the source. Can you click on source there? Get up there. Click on source. Wait. It is computer. Why is it not working? I forgot what's the shortcut to make it work. Now it's on to project it, but it's not projecting.
Is it the correct connection?
Right there, Isaac, in the corner.
Is it the correct connection?
Yes.
I mean the projector, is it based off the correct connection?
Yes. I think it's loose here somewhere.
Try to unplug everything and plug it back in. Would you like any help?
No, it's okay.
Wait, I need to get my things down. There.
Done? Alright, guys! Shh. Now I'm off. Oh, it's coming back. Alright. So, shh. Guys. Please. We only have a few moments again, so we're going to take every single one of these as we can get. By the way, next class that we see each other, we're doing the part B of this. By Sunday, if you're going to do part B, you have to give me the design for the experiment you're going to do. Remember, it's one approved design per group in order to be able to do it. But in order to get the grade for the design, everybody has to have it. So you can do it as a group if one person has a design, right, that I approved. But in order for you to get, you have to do that. So last time we were talking about biochemical pathways which are associated with energy in life. And I talk about how glycolysis is one of the earliest pathways because it has to do with break down sugar. But how glycolysis actually takes an investment of 2 ATP and you end up getting 2 ATP net because you produce 4 ATP total. So that means, and that's actually something important that you need to keep track of for each stage. What you should know for that is where it happens. So glycolysis, where does it happen? Cytoplasm. All right. You should also know that glycolysis takes negative 2 ATP. Right. What else do I need to do it? Well, I need glucose because that's where it starts. Right. I also need NAD. Right. Otherwise, I can't do it because I need to charge the NADs. I'm also going to need ADP because without ADP, right, I can't make ATP. So these are the things I'm going to need. All right. Now, you're actually going to take ATP, but then what you get? So these are my reactants, my products. What do I get? The 2 ATP, right? I'm actually going to use up 4 ADP, meaning I'm actually going to make 4 ATP, right? But then I used up 2 ATP and made 2 ADP, meaning net I only make 2 ATP. Does that make sense? Because, well, we'll talk about nets later. And then glucose turns to 2-pyruvate, right? And then the NADs, 2 of them get made into NADH. They get oxidized, right? Receive electrons. So what's really happening here is that electrons from the glucose are being dumped into ATP and NAD, right? Which means glucose is being reduced. Whoops. Oxidized. Oxidized because you're losing electrons, right? And then you're going to get reduced when you receive electrons. So that means this is going to become reduced. I may have said it backwards last class, so make sure you pay attention to that. When you receive electrons from something else, right? This person who's giving up the electrons, for example, when ATP splits up and becomes ADP plus P, right? ATP is being oxidized because it's losing electrons. You know what I'm saying? So in this case, glucose is being oxidized, losing electrons to charge up NAD and ATP. Right? So at the end of it, you have these 2 pyruvates. Now remember, if you don't have oxygen, right, which is for a long time before photosynthesis evolved, that's what happened, right? All you can do is keep doing this. Now, to keep doing this, you're going to keep having these little nads thing. Now, the cell is going to take care of producing more ATP that's necessary. Because all the ATP that gets produced, the cell's going to use up. Meaning that it's going to have the ATP back. But this one, unless you do the ETC down the road, it doesn't get made up again. Right? You end up with a bunch of these, but to keep doing glycolysis, you need to keep having those. So there needs to be a way to recycle them, right? And that's what fermentation is about. It's about creating, returning that, so that when glucose becomes a pyruvate, right? You're going to make the 2 ATP nets. So you can think about this is a reactant or product, sorry. And then if you talk about net here, talk about what happens net energy. Okay, so that's like, which is what the focus of the thing is. Net energy, I have 2 ATP net, and I also made 2 NAD H. Now, so I made that 2 ATP and the 2 NAD H's. Get out of that. And of course, for that I have to use 2 NAD, right, and 2 ADP. Now the problem is that I want to keep doing this, but to keep doing this, the cell is going to keep supplying back to ADP because cellular processes are going to use this ATP. But unless I do the ETC, I don't get back to the NAD here to charge it. Does that make sense? Meaning that I need to do something to make this happen. And that's what fermentation does. Fermentation gets this pyruvate and converts it to lactate in us. And if it's if you're talking about yeast, it will transform it in ethanol instead. And that's what we use to make beer, to make wine, to make bread, which means bread has some ethanol in it. That solution there has some ethanol in it. Are you with me? Because it's yeast. In this process, the NADH is converted to NADH. So it doesn't actually produce energy. It actually consumes energy. Right? Now, of course, it's 1 glucose to 2 pyruvate. So this happens twice to make 2 lactates to convert 2 of these back to the NAD, which means you can do this again. And of course, the pathway of dealing with the lactate makes us eventually get cramps, but it's not as simple as that. Right? That's what we talked about last class. The next step in cellular respiration would be to do something with that pyruvate and something with that NAD. But before we talk about that, we got to talk about photosynthesis. Why? Because I'm going to teach you in the order that things evolved, right? So let's talk about photosynthesis. Only have 10 minutes. So we're going to, over the next few days, we're going to try to squeeze as much lecture into this energetic stuff as we can. So do not expect to have the entire period to do your lab next time. In fact, I will only give you the last 50 minutes of class. So make sure whatever plan you have in place can be done in 50 minutes. Do you understand that? 50 minutes. Okay? That's all you're going to have. Because at the beginning of the class, I have to do more of this. Is that clear? All right. So what about the key? The key. Thanks, man. Hey. Never mind. You left. All right. So first, I just want to show you a quick surface head to volume ratio. We already talked about it, right? All these folds have to increase that. And of course, if you stack the thylakoids, you're going to get more energy captured. Because if, for some reason, some of the light passes to the first one, the second one grabs it too, right? By the time the light gets to the other side, it's green light because all the blue light has been captured, right? So that's why it's so important, right? If you look at the mesophyll cell structure, you'll see that it's full of chloroplasts. That's what I was showing on here, right? It's actually so they're full of chloroplasts because they're going to be doing photosynthesis. If you look at a plant leaf cross section, right? This is I do my videos. So if you need more than what I'm doing here as a review, please watch the videos I go into. But if you go and look at a plant leaf cross section, you see that it actually has a layer of fat above and below to protect it from water loss and protect it from. Then you have these things here, which is just epithelial cells protecting it also. Then you have this large column-like group of cells. Columns in the Greek temples are called palisades. So that's why it's called palisade parenchyma. And parenchyma is the name of the the ground tissue of the leaf here. All right. So you got the epithelial tissue. Yes. What is this? From yesterday? Oh, this morning I didn't do it. Sorry. All right. So this is the ground tissue of the plant. So you have the epithelial tissue above and below protecting, and then you got the ground tissue here. You also have some vascular tissue, which is also surrounded by a second epithelial tissue, all right, to protect it. These cells are actually important too, because in some types of plants, a stage of photosynthesis actually happens there. The plants which are called C4 plants, they separate the location of where they do the light capture from the Calvin cycle in order to prevent photorespiration. We'll get to that at some point. But what I'm talking about here is that you have the xylem and the phloem. Of course, the xylem is bringing water and nutrients, and the phloem will bring the sugar that's being produced at the leaf, right? Most importantly, though, you see that below the palisade parenchyma, which you might ask yourself, why is it there? Well, the sunlight hits from the top, so this is literally a heat sink. Are you with me? It's just to prevent cells which actually do the bulk of the photosynthesis from getting overheated. Because overheating means problems for the enzymes, that means more photorespiration. So you don't want any of that to happen. So these cells do some photosynthesis, but the primary cells is the spongy parenchyma, which is the mesophyll cells. Now the spongy parenchyma cells have actually a lot of space between them. That's because pockets of air, carbon dioxide and oxygen, which are being exchanged. In the bottom of the leaves is where actually respiration takes place. Now let's think about that. Why would it be at the bottom, not at the top? Because if you open the hole to let carbon dioxide come in and oxygen to go out, right, that's great. But in the stoma, by the way, there's guard cells. And the guard cells, when they're open, right, when they're open, it's because they are not full of water. When they get full of water, if they get really turgid, they close the hole. So we just learned about diffusion. So what do they need to do to get unturgid? They need to lose water. So how do they do that? They pump salt outside. So those cells, if they want to open up, they pump salt outside, and that makes the water follow. Does that make sense? But if they want to close the hole, what do? They pump salt inside, active transport, so that they can get full, and then the hole closes. Some plants have this mechanism of closing the hole in situations where they're losing too much water, right? We're going to learn more about that later in the year, right? But what I'm talking about here, though, is the importance of having that at the bottom. So if you have it at the bottom, the likelihood of, because the problem is the plant can't breathe and lose water, they can't control the oxygen and the carbon dioxide exchange and the water at the same time. So by having it at the bottom, you prevent a lot of water loss from evaporation, right? Because it's going to be at the bottom, right? So the gas don't take to go that way. So it's actually better, you can still breathe, but not lose as much water. Unless, of course, you're a water plant that lives in the surface of the water. Where's all the stoma then? Think about it. It's got to be on the top of the leaf, like a lily. Does that make sense? What if you are a water plant living in the water? Where's the stoma? Well, you don't really have any. All right? Are you with me what I'm trying to say? Because, does that make sense? So why would you have none? Because you're already in the water, so you don't have to, you get it by diffusion, you don't get it to stoma. It's only a land plant problem. Does that make sense? To have this thing? And of course, if you are a very desert plant, are you going to have more stoma or less stoma? More or less? You're in the desert. Less, because you're going to lose less water. No means you're in the water. And the bottom probably means you're trying to conserve. Some plants have on top and on the bottom. Why? Why would you have it on top and the bottom all over the place? Lots of them. Are you worried about water loss? So what place are you probably in? A rainforest. Does that make sense? So the stoma thing makes sense. So, okay. Anyways, back to the actual photosynthesis. So that's why it's spongy, because there's a lot of carbon dioxide exchange going on over there. All right? So, the light reactions will occur inside that chloroplast. Now, if you look at the first part of that system, it's literally a tree of sorts, powered by a solar power plant. And there's actually two power reception disks here, photosystem 1 and photosystem 2. So there's two light recepting disks. Are you with me? What they do is they capture the sunlight to power the rest of the reactions. So in the photosystem 2, there's a molecule called chlorophyll. So if you look at the structure of chlorophyll, right? So you gotta, remember where I left off, review before you come to the next class. So if you look at chlorophyll structure, you look at this thing. You see how it's like it's kind of like a panel, and inside the panel you have the magnesium. You see that? So why in the middle? So all of it is designed to focus the light into the magnesium atom. Are you with me? And then the magnesium gets hit by the light, loses an electron, and that's how the party starts. So the magnesium gets, when you lose an electron, do we get reduced or oxidized? Reduced means gaining a negative charge. But you just lost a negative charge, so that means it's becoming oxidized. So it means he lost an electron. Okay, so what does he want now? He needs an electron. Who's going to give electrons to replace the magnesium's electrons? Water. And that's kind of how the system works. So if you see over here, water gets split by a mechanism that goes way above what we talk about. The magnesium is losing. Are you with me? Chlorophyll, however, is not the only pigment in the surface of the plant. There's actually many pigments. And the pigments all are doing this. But ultimately, all the electrons get dumped into the system just the same. In fact, what actually happens is that, what actually happens is that, let's say, for example, I have to stop here. So remind me, we stopped at the pigments collecting light. All right, we'll talk about it then.
Can I do part C now? Of the last?
Absolutely.
Okay. Because all we have to do is take the video, right?
Yes.